As all of you know, the great Ukrainian geneticist Theodoros Dobzhansky said, nothing in biology makes sense except in the light of evolution, and a corollary of that is nothing in the genomics of energy in the environment makes sense except in the light of evolution. And we've already heard about several beautiful examples. Yesterday, Jeff Dangle spoke about the evolutionary arms race between plants and their pathogens, and we heard about the story from Cameron Curry about ants and their fungi that they cultivate and the bacteria that help them stay healthy. And now we're going to move into a series of talks, starting off this morning with Ashlee Earl, who's going to tell us about whole genome comparisons of Bacillus subtilis.
Thanks. Okay, can everybody hear me? Yeah? All right, good morning. It's wonderful to be here. I've never been to JGI before, so I'm looking forward to touring the facility tonight. So I want to thank the organizers, obviously, for giving me this opportunity. And I'm looking in the camera to thank Roberto for being busy and also giving me this opportunity. So, having said that, I'm looking forward to telling you about some of the work that I've been doing in Roberto Coulter's lab over the past several years as a postdoc, where I've been focused on diversity within this wonderful bacterial species, Bacillus subtilis. And today I'm going to tell you about part of that work. Let's see, what am I interfering with? Part of that work where we've studied genome diversity within this species. All right. So in fear of offending anyone, I thought since I was the beginning talk of the day, I'd start with the kind of the duh question for the meeting. Why is it that we compare genomes? And obviously many of you have answers in your head, but I think for this morning I just want to reiterate that it allows us to begin to ask and answer a number of really big picture questions about organisms, including how is it that groups of organisms evolve? How is it that groups begin to diverge from one another? What are the genome gymnastics involved in that process? I think comparing genomes also tells us a lot about the ecology of a group. What are these organisms seeing out there in the environment? Who are they interacting with? Biotic surfaces, abiotic surfaces, you name it. And of course, and a lot for me as well, it helps us to explain the biological diversity within a group. So why is it, for instance, that one organism or a group of organisms can go in and cause disease and another cannot? So I think it's good to sort of keep these big picture questions in our mind when we're thinking about comparing genomes. Now, I bring up this question, one, to reiterate this, but the other is to segue into the question which some of you may be asking, and that is why compare Bacillus subtilis genomes? And it's a great question, and if I'm going to be completely honest with you, this is my reason. So, what you're looking at here is a number of globally diverse isolates of the species. So I came to Roberto Coulter's lab as a molecular geneticist, and I was interested in biofilm formation. And so what happened, I went out and got all of these globally diverse isolates, and then I spotted them all onto the same exact medium that we use in our laboratory to study biofilm formation, which is a population of cells growing on surfaces. And I saw this beautiful picture. And in fact, Joe Ecker, I don't know if you're in the room, but I just loved your talk yesterday, talking about the Arabidopsis, global diversity. I could totally relate. And so I fell in love with this picture, and the molecular geneticist in me said, why? Why is it that Bacillus subtilis has such wonderful diversity at the molecular level? But of course, I think the questions ecologically, evolutionarily are also really neat, but harder to get at. Okay, but Bacillus subtilis, I think, is a super organism to study in this way for other reasons as well, including the fact that it is such a well-studied organism, at least one strain in particular. And so as a consequence, we know so much about the inner workings of this guy. So you can imagine if you go out into an environment and you find an interesting gene or a gene cluster, in theory, you could put that into the context of the system relatively easily. So I think that's a big strength. Another one is that it's not a pathogen. And I know at least when I started this project, this was a big deal because at that time, most of the intra-species comparative genomic analyses that had been done to that point were focused almost primarily on pathogens, or things that had lived at least part-time in some association within a eukaryote. So I argued that maybe we had sort of a skewed vision of what was going on in genome diversity. And of course, it's apparently it's ubiquitous and it's relatively easy to isolate. So I could go out, I don't have to rely on culture collections, I can get at them myself. Okay, so I started this project, and here I'm going to admit how long I've been in the lab. In early 2004, The nest is very warm. It's hard to leave. But at that time, 454 was just coming into being. I mean, people were just talking about it. And so it wasn't easy to, get your genome sequenced. And so as a consequence, I decided to use as a method to sort of tackle my, what are the molecular reasoning behind these phases. So I wanted to do a comparative genomics approach, right? So I used a microarray-based technique called comparative genomic hybridization. Some people call it genomotyping. Long story short, we had one sequenced genome of the species. We had a microarray that was built based on that genome, and I was able to use that as a platform in which to query lots of different strains, and determine, at least relative to this guy, how much gene diversity was there. All right, so what you're looking at here is a composite of tons of data. So the way it's arranged is that each one of these rows represents a different test strain of the species, and each one of these columns, there's over 4,000, represent the genes in 168, okay? So it's organized, so here's the origin of replication, and as you're going to the right, it's as if you're going clockwise around the genome. Okay, blue, the gene is presumably present in the test strain, and yellow, it's absent. Okay, so I think you already get the take-home message of this slide, and that is my early analyses told me that there was, in fact, perhaps a lot of gene diversity among members of this species. Super, and I can begin to identify which genes may be variable among these populations. But, of course, microarrays are somewhat unsatisfying, right? Because they have pretty severe limitations. We don't know if a divergent signal is really absent, or perhaps it's just diverged significant enough where it can't hybridize. We have no concept of whether there's novel sequences in these strains, and of course if a gene is present, it doesn't mean it's in the same orientation, and these things are all very important. So obviously I'm leading up to the fact that we sequence genomes, and here's my slide, and I'm kind of happy it's here because I feel a little bit burdened by the fact that I'm not a JGI user. I want to be one day, but I think some of you know these guys. They're wonderful guys, Jacques Ravel and David Rasco, who I met and started a collaboration with. It was on the West Coast, which I don't know if that matters. But these guys were then at Tiger, and they were doing Bacillus cereus and Bacillus anthracis comparative genomics. I had some preliminary data in hand at a meeting, and they were on board. So we wrote the grant, and here we are now today. All right, so who did we sequence? So here I'm showing you a phylogenetic tree. It's based on nucleotide diversity among conserved genes. Obviously it's a bit gutted out, but it's just to show you Bacillus subtilis and Bacillus subtilis close relatives who have sequenced genomes in the database to date. So essentially what we did was we sort of filled in this phylogenetic tree, and we chose to sequence strains that are very close to Bacillus subtilis, including close relative Moavensis, Phallus mortis, and then we chose a number of isolates within the species itself. And it turns out that Bacillus subtilis is actually divided into two subspecies called Bacillus subtilis speisizinii and Bacillus subtilis subtilis, for which 168, sort of our reference strain, is a member. And as I hope you can see, is that we're pretty nicely poised, I think, to do an analysis of the evolution of this group as a whole, but I'm not going to tell you about that today. Instead, I want to focus on genomes for which we have full closed genome sequences and belong to the subtilis species. So it includes TUB10, the type strain for this subspecies found in the Saharan desert. This one came from the Southwest Desert, Mojave Desert in the US, and this one is derived from a strain from Germany. All right, so if we just jump in and look at some basic genome statistics for these three genomes, this is what we saw. So there frankly wasn't much striking about them. They're all about the same size, they all have about the same GC content, but if you allow me to highlight the microarray data for these strains, I hope you can appreciate that there's a lot of yellow. So what's going on? Are the genes there and just diverged, maintaining the genome size, or is there something else going on? So we can use our Venn diagram and look to see how much overlap there is among the genes contained within each one of these genomes. And what we found was that approximately 3,000 genes from any given genome were shared by all three. So presumably this makes up sort of the core of what is Bacillus subtilis, and well, the current core of what is Bacillus subtilis, since we're only looking at 3 strains here. But I've already mentioned that 168 has over 4,000 genes, already suggesting, of course, that there are in fact, many strain-specific genes in each one of these genomes. And if we do the math, we see anywhere between 13 to 15% of any given strain's predicted open reading frames are specific to that strain. So presumably, these represent sort of the accessory genes of this group of organisms. All right, so we've got the core genome, we've got these accessory genes. How are they sort of distributed? Right? So what is sort of the architecture of these genomes when we compare them to each other? So this is a graph sort of modeled after the Welch paper showing the E. coli genome analyses. And what I'm showing you is the two different genome comparison. This is Ronnie, and here 168. The x-axis is the genome, okay? And on the y-axis we've got, where did my pointer go? There we go. We've got length of DNA in kilobases. All right, so here's the story. The core genome of these species, or these strains, when the gene is conserved, it's found in the exact same genomic location in the other strain. So there's a high degree of synteny among genomes of at least these two genomes. Okay, no evidence of genome rearrangement. But as you can see, you've got these vertical lines that are intersecting this conserved core. But before I get there, I should just mention that the average nucleotide identity for this core genome, so over 3,000 genes, is really, really high between these two strains. It's over 98%. But once again, let me direct you to these vertical lines that are intersecting this core. And these obviously represent strain-specific islands of DNA in each one of these genomes. So in the case, there's basically two different flavors, as I'm calling it. There's the replacements in red. So these are regions in the genome where it appears that a piece of DNA from one strain has been replaced, apparently, with another piece of DNA from in a different strain. And largely these are unrelated in terms of which genes are there. So these are presumably perhaps hotspots in the genomes of these different strains. And then of course there's the black lines, and these represent what I'm calling, most people call indel. So these are regions that have either been inserted or deleted from a strain since these two strains last shared a common ancestor. Okay. TUB10, 168 comparison, we see very similar results. Highly syntenic core. In this case, expectedly, the average nucleotide identity was much lower, because remember, this guy's in the other subspecies relative to 168. But once again, we've got all of these vertical lines, strain-specific regions intersecting the core. Okay. So what are these strain-specific islands? And I'm sure not surprising to a lot of you here, they correspond to this sort of umbrella term that we're calling the mobilome, right? So these are mobile genetic elements, prophage, conjugative plasmids, insertion sequences, transposases, you name it. And so what I've done here is I've taken prophage that were annotated in the 168 genome, and I've just highlighted them onto these two different graphs I've already shown you. And as you can see, that there's a really high concordance with 168 prophage and the different strain-specific islands that we're seeing in these comparisons. Okay, great. So prophage, phage, appear to be playing a big role in the evolution of these genomes. And of course, we know phage, those of us who study bacterial genomics can bring in a lot of interesting traits that it can allow an organism to perhaps go into a new niche and perhaps begin to speciate. And so in theory, I could take all of these islands, and I can begin to analyze them and get perhaps a picture of the evolution and the ecology of these organisms. But what I'm not telling you up until now is that this is only part of the story. So what I've shown you is only strain-specific islands in these genomes that are 4 kb and higher. But the truth is, if we go below that cutoff and look at strain-specific islands that are 4 kb and smaller, this is the picture that we get. These genomes are literally peppered with strain-specific islands. So you can imagine making a lot of sense out of what's going on functionally becomes a lot harder, especially given the fact that 60% of these guys encode for proteins of unknown function. So we see that it's different, but really, I think the brass ring is, does it matter? And so, in other words, can we begin to identify strain-specific islands that confer some kind of functional, read ecologically relevant change, and do so in a way that doesn't rely solely on genome or gene annotation? All right, so how do we go about this? Some of you may be familiar with phenotypic analysis by Biolog. So we were able to send our strains to this company to do this analysis. Long story short, you take your strain, you put in some base medium, and then you have a number of plates, and within each well on these plates there's some different variable, let's call it. And if the strain is able to grow in the presence of that variable, you get a purple signal. If it doesn't, you don't get a purple signal. And so we were able to score phenotypes for all of our strains, in fact, in over 1,900 different conditions. So we looked at 10 plates of metabolic tests, 10 plates of chemical sensitivity to start to get an idea if these guys are actually really phenotypically different. And to discuss those results, we'll look at our Venn diagram again. And what we found, perhaps once again not too surprisingly to many of you, was that there was a major overlap, right, in the phenotype. So if a phenotype was seen in one cell, or one strain rather, 98% of those phenotypes were shared by all 3 strains. So presumably the ability to grow within these different conditions would be contained within that core gene pool. But obviously you can do the math. Some of them were in fact strain specific. For instance, 168 could grow in a whole host of different tetracycline compounds, and presumably that ability would be contained within that 565 genes that are specific to that particular strain. And sure enough, I can go in and I can start looking at what's there, and I can identify 1, maybe 2 loci that could in fact contribute to that. Similarly, Ronnie had a number of strain-specific phenotypes. TUB10 actually didn't under these conditions, but it did share some phenotypes in common with Ronnie that weren't shared by 168, and similarly, these 2 shared phenotypes that weren't in common with this guy. Okay, so what is this telling us? And from my perspective, I think there's sort of 2 take-home messages here. One, of course, has to do with this overlap. So you get over 98% of your different phenotypes shared by all 3 in spite of the fact that they have hundreds of genes that differ. So what does that say? I think it could say that perhaps a lot of these genes are functionally neutral. They're not really contributing to the physiology of the cell in any important way. Alternatively, maybe we just can't measure it using Biolog. So that's obviously a possibility. The other sort of take-home message that I took away from these data was that there was only one example of strains being able to utilize some new source of nutrition, let's say, and it was these 2 strains could use formic acid, where the other one couldn't. But every other strain-specific phenotype had to do with the ability to tolerate a compound, and largely natural products. So I think that might be kind of neat and might be kind of telling in terms of what gene diversity might be doing out there for populations of this group. And it might suggest that really being able to go into a new niche has more to do with being able to tolerate a new neighbor than it does, using some new carbon source. So it's a limited data set, obviously, but I think it was kind of a neat message to come out. All right, so Bacillus subtilis is not out there tolerating the environment. It's actually contributing biologically to the environment as well. And we know that Bacillus subtilis, 168 at least, can make a number of different chemicals. So here's just the structure of a few of these that are made by these very large gene clusters called non-ribosomal peptide synthetases. And of course, a lot of these have been ascribed antibiotic function. They can kill things. But I just want to sort of bring to your attention, if you're not already aware of this, that there's a really lovely group of literature, I'd say, that's come sort of to light in the last 10 years or so that starts to put a different spin on what these guys might be doing. And it might be that they're not just playing roles as antibiotics. In fact, they could be playing much more subtle roles out there in the environment. And we now know stories where even some of these compounds can actually signal to a different species entirely to cause that particular organism to change its developmental pathways. It can signal to self. And so anyway, not to get too deep into this, but just to make you aware that when we're thinking about the ecology and the evolution of things changing out there, we have to sort of consider them outside of the light of killing other organisms. All right, so I told you they're there, at least in 168, and this schematic is only to show you, not probably kind of poorly, but that when we compare the genome of Ronnie with 168, at least in respect to these big gene clusters, they're all there, they're present, and they're highly conserved, suggesting that they are functioning out there and they're important. When we compare TUB10, it's a slightly different story. And I think it's kind of neat for two reasons. So instead of making this compound encoded by this PPS cluster called plipostatin, TUB10 encodes for something called mycosutolin. And what's neat about this is, one, just the way, it's a little mysterious how this very large gene cluster appears to be so precisely swapped in this genome. There's no real evidence for how this has occurred, but so I think just mechanistically, genome gymnastics is kind of neat. But in addition, let me just show you the chemical compounds. So the two chemicals that are made here, plipostatin and mycosutolin, they're said to be related. I can sort of see that. And they're also said to act against filamentous fungi, so they can kill filamentous fungi. But I think we need to think about perhaps these chemicals in a different light, considering some data from actually myself and others that suggests that the ability to make mycosutolin is widely held in TUB10 and all other Spiziziniae species, or strains rather, whereas the ability to make plipostatin seems to be held within the other subspecies group. So these guys are said, the two subspecies groups are said to be reciprocally monophyletic, meaning that they shared a common ancestor, split, and now they've sort of been diverging. I don't have a lick of evidence, but I think it's kind of neat because this is one locus where it appears that it's widely held in one and the other one holds the other. And so I'm just going to put it out there because I think it's tempting to speculate that maybe this ability to make either plipostatin or mycosutolin has had something to do with that split. Just putting it out there. Okay, so in our these non-ribosomal peptide synthetases, there's other operons in the genomes of these strains that make other antibiotics, and I just wanted to show you these two comparisons at once and bring your attention to these two little operons here called SKF and STP that appear to be specific to the strain 168 but are missing apparently in both of the other genomes. So what's cool about these is that they encode for something that we call killing factors. Now killing factors participate in what is known as cannibalism in these strains. So what cannibalism is you have a population of genetically identical cells, and say for instance, nutrients become limiting. So Bacillus subtilis doesn't like this, right? And just like most populations, when times get tough, generally a subset of your population senses it before the rest, right? That's pretty common. So let's say that this guy here senses nutrients low, and it starts thinking about sporulating, because that's what Bacillus subtilis typically does, right? Times get tough, it sporulates, and it waits for times to get better, or it ends up getting blown across to a better environment. But on its way to becoming a spore, it actually becomes something that we call a cannibal. And what it means is it's making these two compounds encoded by these two little operons. And what happens is its genetically identical brother, who's not quite as hungry as this one, is sensitive to it because it's not making the resistance factor. It causes this guy to be killed. It lyses. Its guts essentially feed its hungry brother. He no longer thinks about sporulating, and he goes back to vegetative growth. And so evolutionarily, it kind of makes sense to do something like this. Making a spore is very energy expensive, and if you can delay it, perhaps you should. So is it that these other two strains rather, are they not cannibals? And so once again, I don't have time to get into all of the different logic behind why I'm saying this, but I think it's tantalizing that there's these two little it's actually little gene clusters in a very similar position as these STP operon in 168 that I believe may be playing an analogous role. And if that's the case, and there's reasons to believe there's some specificity in these guys, so you might have, even though you're not making the proteins, you have some innate immunity, I'm sort of proposing that there could in fact be a sort of unknown until now, really an intra-species war between these different isolates, maybe not unlike the collicin story in E. coli. And I know Peg Riley's here somewhere, and maybe she'll have some thoughts for me on this. Actually, you're right there. I see you. Okay, so my last little vignette, and then I'll be done. And so I've introduced the idea that Bacillus vegetative cell can become a spore, it can become a cannibal, but actually it turns out Bacillus subtilis, and we know this, can become a lot of different cell types, right? So you can have a genetically identical population of cells, and at once, a lot of these cell types will coexist, and it's a beautiful sort of network of signaling and circuitry, and it's really cool, and we in Roberto's lab love this stuff. And so obviously we wanted to know, is there some genetic flexibility in these developmental systems? So let's just go through it real quickly. So we have an empty vegetative cell. We know that it can become any one of these different cell types. They're mostly mutually exclusive. You can't be a spore if you're a swimmer, and you can't be a cannibal if you're making matrix, etc. So there's lots of fine-tuning in the developmental pathway. So we know about what it takes to become one of these guys, the effectors for these. We know a lot about the signals that are actually made within the cell that are actually put out and then sensed by the cell, once again, at different time points to sort of direct when these guys become any one of these different cell types. And then we know a lot of things that go in between: regulators, kinases that pass signals through. There's this Baroque phosphotransfer system. Then you've got antagonists to everything, protein degradative processes, various sundry other things. And we know how largely these are all interconnected. This is actually simplified, I should tell you. And we know their names. And so I can go now and look in my genomes and ask, is there some flexibility? And if so, what's going on? So here's the story. Everything in gray is very highly conserved among these three genomes. And the ones in white are not. Okay, so the message here is that the core guts of this circuitry, this regulation, are highly, highly conserved. So these things are being maintained. But we do see some variation in some of the output, the effectors of these different cell types, including the flagellin, the spore coats. I've already talked about cannibalism. And in addition, we see some variation in the input signals. So some of the signals that are being made and sensed, some of the receptors that sense these things change. And when I say different, sometimes they vary at the protein level, sometimes they're just not even there. So we think that this is probably tweaking the cells in many different ways so that different populations develop differently. And without going into any of the details, but it's kind of a fun story, it's begun to help me answer the question as to why Bacillus isolates have so many beautiful faces. So with that, I'll just summarize, bring back the questions I posed at the beginning. How did this group evolve? Highly syntenic cores. We have lots of strain-specific islands of DNA. The mobilome is a big contributor, right? Phage are playing a big role. When we think about the ecology of the group, this is more largely from the biolog data. We see largely that they're using the same substrates, but they don't survive exposure to the same natural products largely, and there is also some variation in secondary metabolites. That could be very interesting for the evolution of these guys. And of course, to help us to explain at least biological diversity that we're interested in, it appears that the core circuitry is highly conserved, but there is some variation both in the signals and the effectors of these different cell types. So with that, I'll just leave you with my thank yous, and thank you for your attention this morning, and I'm happy to take questions.
Thanks, Ashlee. Questions?
So first, I'd just also like to thank Roberto for being busy, because that was just stunning.
Oh, thank you.
Beautiful summary of a complex area.
Thank you very much.
Second, I just want to mention, I want to follow up on a comment you made about how over the last 10 years people have started to look at these products as presumably communication systems. And I just want to mention the name Julian Davies.
Yes.
I think in this audience it may not be well known, that if you're curious, that's where I go look into
Absolutely.
some of these stories.
He's the hero, the founder of this thought, I would say.
Yeah.
For sure.
And I forgot what the third one was. Oh, I know. So the faces, you didn't get a chance to get back to it, and maybe you and I can chat later, but can the faces have something to do with how they perceive the environment? So for example, those faces may change when you present communication systems to them.
You're absolutely right. If you take those faces and you introduce them to a different medium, some of them change, some of them don't. So I think there's a lot about, so it's not, I posed it as signals that are being self-made, but I think there's environmental signals, nutritional signals, ratios of things that are probably being sensed differently, and they're reacting accordingly to it. So it's a tough problem, actually, to pull apart.